Hey, welcome. Today, we're cracking open a tool that's basically a genetic crystal ball. It's a simple little grid that helps us predict how traits get passed down from one generation to the next, really demystifying the blueprint of life itself. So, let's dive right in.
You've probably thought about this, right? How does this whole genetic lottery actually work? It's all about inheritance, how traits are handed down from parents to kids. But for most of human history, the rules of that game, they were a total mystery.
People obviously knew that kids looked like their parents, but the how and the why of it all was, well, it was mostly just guesswork. A really common idea was that kids were just a simple blend of their parents, kind of like mixing paint. But that theory just couldn't explain how some traits, like red hair, could totally skip a generation and then pop up again later.
And then, in 1905, this simple grid changed, well, everything. It's called a Punnett square. And yeah, it might look like a game of tic-tac-toe, but this diagram, created by a guy named Reginald Punnett, became this incredibly powerful way to visualize and predict the probable outcomes of a genetic cross. It's our crystal ball for heredity.
So, how in the world does this thing work? Let's build one from the ground up, okay? We'll start with the simplest case, tracking just a single trait. This is what's called a monohybrid cross. But before we actually draw the box, we got to get on the same page with a few key terms.
All right, first up, the allele. The easiest way to think about an allele is like this. If a gene is a category, say pod color, then the alleles are all the different options within that category, like green or yellow. And just as a heads up, we usually show the dominant alleles with a capital letter and the recessive ones with a lowercase letter.
Next up is the genotype. This is the actual genetic code an organism has for a trait, the specific pair of alleles it's carrying. Since you get one allele from each parent, your genotype is always going to be a two-letter combo, just like the GG you see here.
And that genotype is what determines the phenotype. The phenotype is the physical trait you can actually see. So, for example, if the genotype is GG and that green allele is dominant, the phenotype, what you see, is green pods. Think of it as the genetic code versus what that code actually builds.
Actually building the square is a super straightforward four-step process. First, you figure out the genotypes of the parents. Second, you split their alleles up and put them along the top and the side of the grid. Third, you just fill in the boxes by combining the letters from the top and side. And last, you analyze what's in the boxes to see the probable outcomes. Simple as that.
Let's watch it in action with a classic pea plant example. We're going to cross a parent that's heterozygous, which just means it has two different alleles, a big G and a little g, with a parent that is homozygous recessive, meaning it has two identical recessive alleles, G and G. See? We just split the parent alleles, pop them on the grid, and fill it in.
And voila! The results give us the probable outcome. By just counting up the boxes, we can predict that there's a 50% chance the offspring will have the GG genotype, giving them green pods, and a 50% chance they'll have the GG genotype for yellow pods. Now remember, the Punnett square doesn't tell us the definite future, but it gives us the odds.
Okay, but here's where things get really fascinating. Genetics isn't always so black and white, or, green and yellow in this case. Sometimes the genetic rules get a little blurry. And that brings us to this really cool concept called incomplete dominance.
So here's the key difference. In all the examples we've seen so far, one allele completely dominates the other. But with incomplete dominance, when an individual is heterozygous, has two different alleles, it shows a phenotype that's actually a blend of the two. It creates something totally new.
The absolute textbook example of this is the snapdragon flower. So check this out. If you cross a red snapdragon with a white one, you don't get some red and some white baby flowers. Nope, you get all pink flowers. Neither the red nor the white allele is fully dominant, so they just kind of meet in the middle. Okay, so we've totally mastered tracking one trait, but what if we want to get ambitious and track two different traits at the same time? Like maybe both seed shape and seed color? Well, for that, we're going to need a bigger box. This is what we call a dihybrid cross.
Now, I know this might look a little intimidating, but trust me, the principle is exactly the same. We're just using a 4x4 grid now to track all the possible combos for two different genes. You figure out all the possible allele combinations each parent can pass down, you put those along the top and side, and then you just fill in the grid like before.
And what's amazing is that when you cross two parents that are heterozygous for both traits, a super predictable pattern emerges. You almost always get this famous 9:3 to 3:1 ratio in what the offspring look like. 9 will show both dominant traits, 3 of each mixed combo, and only one lonely little guy will show both recessive traits.
All right, let's add one more layer of complexity. What happens when a trait is carried on the very chromosomes that determine biological sex? This is where we get into sex-linked traits. And believe me, the patterns of inheritance get pretty interesting.
So, as you probably know, biological females typically have 2 X chromosomes, XX, while males have 1 X and 1, much smaller, Y chromosome. Now, because the X chromosome is so much bigger and has way more genes on it than the Y, most sex-linked traits are specifically X-linked. They hitch a ride on the X.
And here is the absolute crucial point. For a female to show a recessive X-linked trait, she has to inherit 2 copies of that recessive allele, one on each of her X chromosomes. But a male, he only has 1 X chromosome. So if he inherits that single recessive allele on his 1X, that's it. There's no second X chromosome with the dominant allele to mask it.
You can see this powerfully illustrated with conditions like hemophilia. So in this example, you have a carrier female. She doesn't have the disorder, but she carries the recessive allele, and she has children with an unaffected male. The Punnett square shows that there's a 25% chance of having a son with hemophilia, but a 0% chance of having a daughter with the condition, at least in this specific cross.
So as we've seen, this simple little square is an incredibly powerful and flexible tool. It's really a cornerstone of any geneticist toolkit, allowing them to predict all kinds of complex inheritance patterns.
So let's do a quick recap. We've used the Punnett square to track single traits, 2 traits at once, those cool blended traits from incomplete dominance, and even traits that are linked to our sex chromosomes. But the Punnett square has one more clever trick up its sleeve. You can use it to work backward and solve for an unknown.
This is called a test cross, and it's basically genetic detective work. Let's say you have an organism and it's showing a dominant trait, but you have no idea if its genotype is homozygous dominant or heterozygous. You can solve the mystery. You just cross it with a homozygous recessive individual, and the results you see in their offspring will tell you exactly what that unknown parent's genetic code was. From Gregor Mendel's humble pea plants all the way to modern genetic counseling, the Punnett square has been absolutely fundamental to how we understand heredity. This simple box gives us the incredible power of prediction. So the final question is, as our ability to not just read, but to write genetic code advances, what will we choose to predict? And maybe, what will we choose to design?